and thanks for joining us today. My name is Abby, and I'm one of the organizers of this year's Quantitative Seminar Series for the Converse Lab. And I'll just begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So today's presenter is Dr. Ben Augustine. Ben Augustine is a quantitative ecologist working as a postdoctoral scientist for the United States Geological Survey. His recent research focus has been developing models and MCMC methods to estimate abundance via capture-recapture and occurrence via species occupancy in the presence of uncertainty in individual and species identity, particularly in the context of non-invasive sampling methods, as well as data processing pipelines involving machine and deep learning classification methods. So Ben will take questions after the talk. And thanks again, everyone, for coming. And then big thank you to our speaker for joining us today. And with that, I'll turn it over to you, Ben.
Okay, you can see and hear me and see my screen?
I can.
Okay, and gonna hide the control panel here. And okay. Yeah, thanks for having me, and thanks for setting this up, Abby. And I think Brielle as well. And again, I'm Ben Augustine. I'm going to be talking about some of the research I've been doing over the past 6 years or so as part of my PhD and postdoc work. And today I'm going to be talking about the utility of using spatial information to improve individual classification in ecological models of abundance. And you'll see more specifically, I'm just talking about latent identity spatial capture-recapture models, but I'm going to be kind of focusing on the classification angle, classification of individual identity. Okay, so in what sense is classification relevant to ecological models? Well, the relevance is that probably most ecological models require highly accurate classification of samples in various senses. So, for example, in capture-recapture, we need to be able to classify the individual identities of each sample. And in occupancy models, we need to be able to classify the species of each sample. And when we make errors in these classifications, it introduces bias into the ecological parameters that we care about. And not many errors can lead to quite a bit of bias. I'm just reporting couple of simulation studies here for capture-recapture. 1 to 5% ID error, in a particular model. In that case, led up to 10 to 20% bias, and we see similar results for occupancy. And these are simulation studies, so it's a theoretical exercise, but I think it's uncontroversial to say that inaccurate classification is not uncommon using even established sampling methods that we use in ecology, particularly non-invasive methods such as genetics, remote cameras, and bioacoustics, but even other methods. But in thinking forward, another consideration is that new tools are emerging to help us handle big data, whether this be hundreds of thousands of camera trap photos, massive data sets of aerial photos, or thousands of hours of remote audio recordings. And we need statistical models that explicitly allow for classification error, so we don't so these big data methods don't leave us with big bias and incorrect inferences. So now I'm going to give some concrete examples of individual classification problems that arise in non-invasive capture-recapture type data. And these models were the main goal is estimating density, but also other population parameters such as survival and population growth rate. So first, a widely used method to collect capture-recapture data, one of the most established, is the use of camera traps for individually identifiable species, particularly species where you can visualize their flanks. And one way to think about this is we're using human classifiers to assign these photos to individuals. And the status quo is assumed that we can do this perfectly, but studies rarely, almost never provide evidence to support a negligible error rate. And in a recent study of captive snow leopards, compared identity assignments across multiple observers and found that those humans misclassified 12.5% of the photos, or I think they disagreed on 12.5%. You know, and if they calculated, they would have overestimated abundance by 1/3. Now, one study doesn't, you know, tell us much about how common that level of misclassification is in this, you know, well-established approach, but I think it at least it indicates to me we should be taking classification error more seriously, even in approaches that we have kind of been ignoring it so far. So another established method is the use of genetic markers, and traditionally we use microsatellites, but more recently we've been using single nucleotide polymorphisms, or SNPs. And the basic idea is that if we observe enough genetic markers, two individuals will very rarely have the same complete set of markers by chance. We call this multi-locus genotype. And to assign an identity to a genetic sample, say a scat sample, we amplify the DNA, then use a process of human interpretation to classify the true values of the genetic markers. Or more recently, people have been using error-tolerant matching. But observing the genetic markers is complicated, particularly because of genotyping error. You know, and this is widely known, and to limit errors, typically there's rigorous error checking protocols, and these can produce very reliable data, but they can't guarantee all errors will be removed, and it often requires discarding a lot of data. So here's an example from autonomous recording units, or ARUs. And methods for identifying individuals from spectrograms of audio recordings are relatively new, but they have the potential to be used for capture-recapture abundance estimation if we can deal with uncertainty in individual identity. Here's a spectrogram of a given call from this Klink et al. Paper, where the frequency is on the y-axis and time is on the x, and you see these notes followed by a trill. And Klink et al. Were able to use linear discriminant analysis to classify calls to individuals using the durations of these notes and trills. On the bottom right here, we see the first two discriminant functions with 90% confidence ellipses around these different individuals. And we see some are well distinguished from the other ones, but there's a lot of overlap among other individuals. And of course, you add more of these more dimensions, you're going to get more differentiation between individuals. But say if we're just using these two dimensions of the data, they would, if we were to assign these to individuals, there would be quite a bit of error. Moving back to cameras, we could also produce individual identities using morphometric measurements. On the left is some measurements made manually by humans of bison from this Merkel important paper. And on the right is this bear ID project. These are the bear morphometrics are harder to measure than the bison, particularly because we have a bunch of different photo angles. So the bear ID is using deep learning to extract morphometric features and then classify these classify the photos to individuals. You see here, these are the measurements they're interested in, and here are what they call the embeddings. And you know, this is basically just another data reduction, just like for these bioacoustics. We've got the individuals grouped. Some of them are well separated, but some of them overlap quite a bit. But I think it's interesting that both of those two methods leave us with kind of the same exact uncertainty in individual identity. All right, so I'm going to move on to how we're going to incorporate this uncertainty in individual identity into the capture-recapture models we want to use. So we're going to use what I'm going to call couple classification, and I get this from the Carey and Royal AHM2 book. And the main idea is we couple the classification process, the classification model, and the ecological model together to jointly estimate the parameters of both and propagate the uncertainty bidirectionally. And this can be contrasted with, an uncoupled approach where it's a two-step process where you classify samples. You've got some sort of confidence threshold, and samples that don't meet that threshold, you discard. Then you construct a capture history with all the samples that pass the threshold, and you assume that it's perfect, and you fit the ecological model. So what we're going to do is to classify and fit the ecological model simultaneously. Okay, so using coupled classification, we're going to recognize that the classification process is just one more process that determines the data we observe. So therefore, we just kind of tack it on the end. We've got the ecological process, which might be what determines density, etc. The encounter process, and then the classification process of all the samples we encounter. And this has been done before in a, more or less in non-spatial capture-recapture. Here's two examples. The latent multinomial has been used in many different applications and generalized and extended to do more things. And then we also have this write it off paper for genetic classification. All right, so here I'm going to transition to the importance of using spatial information for classifying individual identity. So first, why does the spatial location of encounters give you information about individual identity? Well, very simply, it's because individuals use different parts of the landscape. A lot of species have well-established home ranges, or just during the time you're sampling, some animals are going to be in some parts of the landscape and some others. So the basic idea is that if you observe, say, a sample here, it's more likely to belong to an individual that lives around here than it is one over here, given that space use is fairly restricted. But if space use is large to your area of interest, then spatial location gives you much less information about individual ID. And so to capitalize on spatial information, we need a model, a statistical model that has all these features of the number of density of individuals, that's spatially explicit where they live, and the extent of individual space use, and spatial capture-recapture fits the bill. So what is spatial capture-recapture? I got these slides from Cyril Milleray on the NEE blog. This is a nice, concise description. Basically, it's just a model it's a spatial model for density and detection. So there's a model for density and individual location, and these individual locations are represented by activity centers. Then there's then the extent of individual space use is determined through the spatial scale of the detection function. With this spatial scale parameter sigma, determines how far away from an activity center that an individual can be detected. And here's a hypothetical realization of the SCR encounter detection process with 4 individuals here, and you can see the spatial dispersion of their captures governed largely by sigma. Looking at this in a little more detail, a spatial capture-recapture model specifies a two-dimensional state space, so basically the area our population lives in. We have X, which is the coordinates of detectors. I've got these represented with these little X's here. We'll sample for K occasions, and it produces Yij. These are the data, the detections for individual I at detector J over K occasions. You can also model this Yijk if you have occasion effects. Okay, so for the ecological process model, here it combines models for abundance and individual location. And the model for abundance is typically induced by a data augmentation process where we, I won't describe in great detail because I believe everyone here probably is familiar with it, but basically we append all zero capture histories to our data matrix Yij, and then also associate a Z with each potential individual. And if we assume Zi is Bernoulli distributed with parameter psi, this induces a binomial relationship on binomial model for abundance. With M is just how much we've augmented, and psi is the probability that one of these Z's is on. And then we've got a model for the distribution of activity centers across the state space. And here I'm just assuming this is uniform. There are many other options. And these Z indicators, basically during the MCMC process, we're going to be turning those on and off to determine how many of these are in the population. And so here I've got one's grayed out would be off, one's black turned on, and if you capture, they must be turned on. And for the observation model, we have many options. Because we're going to do latent ID, I'm just going to show the Poisson observation model here. And basically, we've got the detection function here where lambda ij, the expected number of detections for individual I at detector j on one occasion. And we've got a baseline encounter rate in the spatial scale parameter. And we just assume that our observations are Poisson, lambda ij times the number of occasions times this z. So if the z's off, it's going to be Poisson 0 with all zeros. So this completes a very simple spatial capture-recapture model. And here I was just showing these are realizations of the posterior for the latent structure. Basically, we just have to estimate the activity center locations. You see the Z's coming on and off. So the latent variables are the Z's and S's. That's a basic spatial capture-recapture. All right, so back to this classification idea and why spatial information is important. What if we don't know the individual IDs in an SCR model? Well, we're going to observe a spatial pattern of detections whose locations can be regarded as partial identities, or some sort of individual feature measured with error, where this feature is the activity center and the observation is where the detections are. So that would be the true feature is an activity center. We're observing it with error around its activity center. And so this is essentially a cluster analysis problem with an unknown number of clusters, and it's strikingly similar to the previous bioacoustics and morphometrics classification problems. You know, in all cases we've got you know, two dimensions of measurements, and we're trying to figure out how many clusters there are. So it's more or less the same type of problem. Okay, so now I'm going to show you the most basic latent ID SCR model that uses spatial information only to classify individual identities. And that is Unmarked SCR, model from Chandler and Royal. And this is exactly the SCR model I just described, except now the individual identities are not known. So basically, we know the traps and occasions of captures, but we don't know the individual identities. And you can fit this model either by updating the latent capture histories or equivalently the latent IDs, or marginalizing over individual ID in the observation model. However, if you want to use, so you can marginalize in the Poisson. However, if you want to use any other observation model, you need to update the latent capture histories or the IDs because you can't marginalize over ID in the observation model. But with the Poisson observation model, you know, these trap level counts are also Poisson with a rate parameter, which is you just sum over I of the encounter rates for the individuals by traps. and the inclusion vectors multiplied by k, and I should have a j index here and here. All right, so let's see. Yeah, so if you're using a Poisson distribution, just some interesting facts here is the full conditional for how you would distribute the samples that are trapped is multinomial with this rate for each detector. Equivalently, the full conditional for ID is categorical for sample L collected at detector J. So that's just one member of this multinomial count. And so as far as latent variables, we still have latent activity centers, latent Zs, but we also have latent IDs. So now we see there's switching of these connections of samples to activity centers. So these are posterior realizations from unmarked SCR. Okay, so next we're going to add categorical identity covariates to unmarked SCR, and I call this categorical SCR. And these categorical covariates could be genetic loci, they could be sex or age class that you might be able to see in a photograph, or anything else. And for this model, we just append categorical features onto each individual. So each individual has some true categorical features across M covariates. So there could be, say, M genetic loci. Each individual has a true multi-locus genotype, where they have a true sex and an age class, etc. And when we capture them, this model assumes we observe it perfectly. So the observation of sample L at covariate M is just whatever the true value is for the individual of that sample L at that covariate. Although you can also have missing data. You might not be able to, not all loci might amplify, or you might not be able to tell the sex of an animal in all photos. So now the full conditional for ID is exactly as it was before, except we exclude matches that conflict with, we remove, conflicting samples that cannot match. You observe values, they're inconsistent with the truth of these individuals. So basically you can just zero out matches that were impossible. So for example, if you observe a photo as a male, you cannot match it with any females, etc., because there's no possibility of observing those category levels with error. All right, so I wanted to make a quick point with this model that you can achieve practically certain individual identity through partial identity. The idea is that you add more partial identity covariates, you're incrementally removing uncertainty in individual ID until none remains. And to show this, I'm going to show just a quick simulation study using 0 to 8 categorical features with 2 levels each. So we could say these could potentially be SNPs, because SNPs have 2 levels. So if you have no covariates, that's unmarked SCR, and then we're adding 1 through 8, and the number of unique population groups where their IDs are consistent with each other scales by 2 to the k. So with 8, we're up to 256 unique identity groups in the population. All right, so at the top, I'm showing a histogram of the posterior modes across 100 simulated data sets as a function of the number of unique population, unique identity groups, or number of SNPs, hypothetically. And on the bottom, I've got the mean 95% credible interval width as a metric of precision. And so this is unmarked SCR, and it's got, it's very inaccurate. There's a lot of sampling variance, and it's not very precise. But as we start adding more and more partial IDs, we're squeezing out uncertainty in ID, and we're converging to the SCR estimate of perfect ID. See, and you see, you get diminishing returns as you start. Once you get a certain amount, you're just getting diminishing returns because there's hardly any uncertainty left. And so to me, this illustrates there's many intermediate levels between unmarked SCR and perfect identity SCR. And we don't require a perfect identity. And preferably, we would like to be in this zone and not this zone. So more partial information is better. And this is an example with categorical covariates measured without error, but this same thing happens using continuous covariates measured with error, or categorical measured with error, or many other possibilities. If there's measurement error, generally you just need more of them to achieve the same precision and accuracy. So now I'm going to walk through a paper. It's kind of the best case study that we've got so far, where we jointly classify microsatellite markers measured with error to individuals while estimating population abundance and density. All right, yeah, so yes, stepping back a little bit, one of the most important tasks in genetic analysis is to assign individual identities to the samples. And this is often called genetic fingerprinting. What does a genetic fingerprint look like? Well, it looks like this. This is an example of a 7 loci genotype using microsatellites. And for microsatellites, each of these loci has a certain number of alleles. Each one has 2 copies, and each allele is the number of sequence repeats. So this would be 158, 158 sequence repeats. So both copies would be the same. Where here we've got 231, 245, and there the 2 copies would be different. So multiple individuals in the population will match in any one locus, but the idea is that if we use enough of these, then there won't be 2 individuals in the population that share the same multi-locus genotype. When that happens, it's called the shadow effect. And basically, you just need enough loci where that's rarely going to happen. But you can never guarantee that it didn't happen. So given estimates of the allele frequencies for a set of genetic markers, you can calculate PID with the probability that 2 randomly selected individuals would share the same multi-locus genotype by chance. This is just a measure of the discriminatory power of the marker set. So basically, you want enough markers where you've got this very low. And in practice, PID SIB is used because individuals in the populations are related. They could potentially be siblings. So that would be the probability of 2 randomly selected siblings having the same multi-locus genotype. Yeah, so I believe that's all I wanted to say on that slide there. But the main challenge for using genetic markers from non-invasive samples is that the DNA is often of low quality and quantity, and these lead to partial genotypes where certain loci don't amplify and genotyping errors. And if you have a rule that you have to use 7 loci to achieve a very low PID, and you start not observing them, you're not going to be able to use these samples. So if we don't observe this locus, but the sample matches this one at all but this one, it's probably this individual. We just can't be completely sure. And this one, maybe it's that same individual, maybe not, but this is definitely consistent with that individual, and we would like to use these samples. And then the other problem is genotyping errors, which the kind of way that they're typically classified is you have allelic dropout, which is when a homozygote here, or heterozygote, sorry, 231, 245, it's got 2 levels, heterozygote. If one drops out, you observe 231 only, and that's allelic dropout. And that's the most common type of genotyping error. And then false allele is basically anything else. But these are typically very rare. but if the DNA quality is extremely poor, they might not be. All right, so again, what's typically done is there's often some sort of replication and scoring rules to generate consensus genotypes, although there's some protocols that don't use replication. Basically, they've got some rule. You need to see a homozygote a certain number of times, say 3 times before you're scored a consensus. If you see it once, maybe you're not going to be able to score it. And yeah, the basic idea is that the geneticist is going to take all this replication and generate the data that we're going to input into the statistical model, but this is the actual data for the classification task. And so we're going to use that instead. Okay, so here is a broad overview of this model. We've got, this is a couple classification approach with an ecological process, capture process, genotype observation process. Ecological process is going to tell us how many individuals, where do they live, what are their genotypes. Capture process tells us where and how often individuals are detected, given how many there are and where they live. And genotype observation process is just which genotypes we observed, given where they were collected and which individuals those were. And so here's the ecological process model. It's a joint model for these individual genotypes and activity centers. So we've got genotype frequencies, which we're going to estimate. We're going to estimate the true genotypes, the activity centers, and we've got some prior for the spatial distribution of activity centers, though there might be a model with parameters of this inhomogeneous density and a model for N. And because individuals all have their own spatial location, they can have the exact same true genotype. So the shadow effect, where 2 individuals have the same genotype, would be allowed in this model. It is allowed. The capture process is typical SCR observation model, except we don't observe an ID, we observe a genotype. Let's see, and so this is like an example of a transect search. The only difference here is often individuals don't have, well, I would say probably always, they don't have the same home range size. So we're going to use a model for individual heterogeneity and detection. And I'm going to use this compensatory model from Effort and MOAT. Basically, if we reparameterize it in terms of A0 and sigma, where A0 is a single detector effective sampling area, we put a continuous individual random effect on sigma, and that induces one on lambda. So lambda naught. So basically, if sigma gets bigger, lambda naught goes down. And so it's assuming space use, the detection is proportional to space use. In our genotype observation model, we're going to allow allelic dropout false alleles, and we're going to have sample quality covariates. And you can use continuous covariates where it might be, say, DNA symmetric of DNA quantity or quality, but that is much more computationally intensive than just kind of splitting them into groups. So we're going to say there's high quality samples and low quality samples, and that's just determined by how much failed amplification there are in the first 3 reps. So if you see, I forgot what the exact, I think I used, if you observe 8 or more of those 9 loci across the first 3, it's a high quality sample. And if you observe less than that, it's low quality. And that more or less matched up with the samples that geneticists said were reliable and the ones that they said were not reliable. So just kind of a note about this model as an unsupervised classifier. One way to look at this is the full conditional from Unmarked SCR, and that's functioning as a prior for the sample ID, and we call this like an ecological prior. And this is coming from the ecological and observation model. And then we have the genotype observation that's giving us information from ID as well. So if you were going to use the genotypes in classifying with, say, like a finite mixture model, you would need to specify some prior for ID, which might be uniform across all possible IDs. But now, because through the ecological and observation model, we have kind of a smart prior that is incorporating the ecological information. And so, yeah, the ecological prior is basically telling us how consistent is that sample location where we found the genotype with an individual eye. And the genotype observations are telling us how consistent the sample is with the genotype of individual eye. And then I'm going to, just walk through this quick application of fissures in New York. So in this study, there were 608 hair snares, 420 samples, and of those, 263 were identified to individual, by geneticists and used in the original paper by Linden et al. And so that leaves 105 samples. There were partial genotypes. There, some genotype information there. And then 52 that completely failed, but they still have a spatial location, so we can still use them. And there were 3 to 7 PCRs, or replicate genotypings. And in this exercise, we're just going to compare the original SCR, an analysis of the original SCR data set with this new model using all the samples, and to see if we can improve our estimates of the ecological parameter of interest, which is just abundance or density. And this is a typical high quality sample where you get a lot of good replication. This is a typical low quality sample. You've got a lot of failed replication. And, if this was the same individual, this is 202, 204, and this one's 202, 202, 3 times, that would imply you had allelic dropout 3 times in this sample. And also allelic dropout here 2 times in this sample. So this is consistent with that one, but we're just not observing enough information to be very confident. Okay, so here are the parameters we're going to look at. These are the parameter estimates for the SCR model. And we've got the single detector effective sampling area, spatial scale parameter, the variance of the spatial scale parameter, how many individuals we captured, which is known in SCR, assuming we made no errors, the abundance estimate. I've just got spatial recaptures here. There were only 9 in that data set of very certain ID. But when we add all these discarded samples, the individuals are more detectable because we're just using more samples. And we detected a lot more individuals. We're not sure exactly how many we detected, but we can produce a posterior report, and it's almost certainly more than we detected in the original data set. Spatial scale parameter is estimated more precisely, as is the variation. And we get a posterior for the number of spatial recaptures. So there's a lot of uncertainty in how many spatial recaptures there are, particularly because of the samples with no genetic information at all. But we're quite certain we've got more spatial recaptures than we did originally. But the key metric here is this: the abundance estimate was about 27% more precise, and the point estimate, it's a tad higher, but it's consistent with the previous one. And the identity assignments made by this model almost perfectly match those made by the geneticists in this data set, except there were 3 cases where it looked more plausible that the geneticist may have made an error, but it takes a lot of investigating to kind of figure that out. And here's some of the some of the pairwise match probability. So you can calculate the posterior that sample A belonged to the same individual as sample B, and these are the low quality samples. Some of them, 13, were matched to another sample with a probability of over 0.95. So have a lot of information about individual ID, 28 samples over 0.75, and then 60 samples over 0.5. Just the genotyping error rates for per replication. So I don't have the false allele rates on here because they were both very rare for both high and low quality. But for high quality samples, the probability you're going to score a locus correctly was per replication is 0.8, and probability of allelic dropout was 0.2. These low quality, it was about 50-50. So as we expect, there's a high error rate, but we're still extracting a lot of information about individual identity because of our model that allows for this classification. And what's cool about this is we can interrogate these spatially explicit pairwise sample matches. So these are those two previous samples. And the model matched this sample up with this candidate where they're inconsistent here and here, but it just implies that if this was the same individual, this was allelic dropout 3 times, allelic dropout 2 times. If the allelic dropout per rep is 0.5, it's not that unlikely that this sample belonged to this one, but it still seems like 0.99 is a very certain probability to assign to that match. But when you consider the spatial information, here's the incomplete study area. Here we're zoomed in on where these two samples were right next to each other. Here's kind of the approximate space use of Fishers. And in this little region, there's probably only 4 or 5 possible individuals that were there. And so, the low population density combined with the very close spatial proximity of the samples is what's contributing to this high probability match, in addition to this genotype information. Okay, so in summary for this model, we've got a single model for assigning individual identities to these genotyping replicates while estimating the population parameters of interest. This is improving the genetic classification, but it's also improving the estimates of the ecological parameters that we care about. And the uncertainty is being propagated bidirectionally, and there's no data screening, so we're not discarding data, and we have clear assumptions and probabilistic relationships, which is contrasted with the approach of using decision rules for the scoring of the multi-locus genotypes. And yeah, I didn't make a slide for like the limitations of these methods, but I would say 1, they're computationally intensive, and 2, they require more effort to get the capture-recapture assumptions closer to the truth, particularly things like variability and home range size. Because if you have a lot of variability in home range size, and you're going to underestimate home range size, and individuals with big ones are going to be split into multiple individuals. And so that's just one example of there's more, basically there's more sensitive to violating model assumptions. But as you get more partial ID information, they become more robust to violating the assumptions because they're getting closer to SCR. So the maximum robustness available is what you would see in SCR. And so if you have very sparse information, partial ID information, the model is going to be more sensitive to assumption violations than you have very abundant information like we have in these genotypes here. Yeah, I think that's all I was going to say about that. And since we have time, I'll just kind of show this kind of landscape of current latent ID SCR models. And we've kind of got a mix and match here. We've got the base models, we've got unmarked SCR, where we don't know any of the individual IDs for sure. We've got the random thinning model, where individual IDs are available for some samples from all individuals. We have spatial mark recite, where we split the population into 2 groups, one of which we can get individual identities on, though we don't always. And another group, the unmarked animals, where we can never get that. We might know the number of marked individuals, like we pre-mark them. And we get one categorical partial ID covariate, which is marked status. So if you know a sample is unmarked, it can't belong to a marked individual and vice versa. And yeah, and then spatial capture-recapture is not a latent ID SCR model, but the random thinning and spatial mark recite are kind of intermediate cases between unmarked SCR and spatial capture-recapture. And then for any of these, except spatial capture-recapture, where it wouldn't be relevant, we can add partial identity information. They all have spatial locations. We can add categorical ID covariates to any of these. They can have observation error. They could be continuous with observation error. They could be Dirichlet vectors like you get from deep learning, etc. They could be pairwise matches, like this flank is the same individual as this flank, and you can model uncertainty there. These are the combinations that have been published so far. And then, yeah, multi-method SCR, which is kind of the same model we use for the flank matching, is more general, where you can probabilistically resolve identities across inconsistent data sets, like a photo data set and a genotype data set, etc. So that's all I have. And one of my postdoc funder for the work I did on this as a postdoc with the Acknowledgements Center at Cornell. There's some selected co-authors, and I've got a lot of examples on GitHub in Nimble. So that's that. I'll take questions.
Thanks so much, Ben. That was fantastic. I've got a couple of questions, and then that'll give folks online a chance to either raise their hand and I can unmute you, or you can type your question in the question box and I'll bounce that over to Ben. But to start off, so really early on in your talk, you talked a little bit about data augmentation as opposed to reversible jump MCMC. And I was just curious if you could talk a little bit more about, like, what are those two doing differently? When would you use one versus the other, and kind of what are the pros and cons there?
Yeah, well, I just recently stumbled upon reversible jump in spatial capture-recapture because I don't know if you've seen the Robbie Emmett's group SCR paper, but there we used, we called it double data augmentation. Well, I called it that, and I think it's actually, I was actually using reversible jump for the, so you've got data augmentation there for the groups, and then data augmentation for the number of individuals per group. But I realized I was actually using a process that was closer to reversible jump to retain the assumption that, N is Poisson. In that case, number of individuals per group, well, there is 0 truncated Poisson. But you can use that same approach in spatial capture-recapture, and it's so I guess the main difference is reversible jump, as typically described, doesn't augment the data. You're dynamically moving the dimensions. When you add an individual, you add an activity center, and you just delete them. So the dimension of the data and the latent structure is always changing. Whereas with data augmentation, the maximum dimension is fixed. So that's one distinction. And so reversible jump is a lot faster than typical data augmentation. However, using data augmentation, if you can make it practically as fast as a reversible jump if you skip calculations when the z is 0. When the z's are off, if you don't calculate like the distances from from that individual to the traps and the expected detection rate and all that stuff, and you can also not update the latent or the activity centers when z's are off. And so, and then with the final step of not updating every Z on every iteration, so reversible jump, you're saying you're going to have a proposal, say N's currently 50, you might propose between 45 and 55, and accept or reject that in a Metropolis Hastings step. So you're only proposing to turn on or off, up to 5 Z's at a time, whereas in data augmentation, you go through every Z and try to update it, and that's generally inefficient. So if you then only update a certain number of Z's on every iteration, then they're pretty comparable in terms of speed that I've seen so far. And there's even like hybrid ways where you can do reversible jump with pre-allocation, which I find useful to make it work in Nimble, because Nimble's not going to like if the dimension of the data is changing like that. And I've gotten some pushback saying, is that really reversible jump if you augment? But I think the main distinction is reversible jump. You don't induce. You can retain like N Poisson lambda assumption, where as data augmentation, you're inducing the N is binomial M psi. That's the main distinction, in my opinion. Anyway, does that answer your question?
Yeah, no, that helps a lot, and it kind of leads into my other question, which was, I do most of my work in Nimble as well, and I've explored writing custom samplers and stuff. So I'm just generally curious about how you get these things to mix and sample efficiently. If you could just speak to that a little bit.
I think it's problem specific. Let's see. So regarding mixing, the hardest part to get good mixing in a SCR model with latent IDs is to get a good activity center sampler. So when I was originally working on these models, before I switched to Nimble, I would just set one. So typically those activity center updates in Metropolis Hastings, I would set one tuning parameter for all the activity centers, and it's pretty, like, it's pretty easy to know, ballpark roughly what that should be. But then you can check your acceptance rates and see if you need to change it. But to make it more user friendly and moving to Nimble, it would be nice to have Nimble tune those activity centers itself. So that's been the main challenge, is coming up with smart activity center samplers, because the data associated with any activity center is constantly changing. And so the proper tuning parameter, when you have, say, 8 samples allocated to an activity center, it needs to move less than if it has 1, it's going to move more. And so the best you can do is kind of try to tune for what's good on average, and just kind of through trial and error come up with kind of decent strategies that seem to work well in most of the cases I've considered. But that's generally the hardest part regarding mixing. As far as efficiency, just trying to code everything as efficiently as possible. Yeah, does that answer the question?
Yeah, that helps a lot. Thanks.
I mean, my current best idea, I think, for tuning the activity centers that worked well in Nimble is to I set it up so that it only tunes the activity centers when the Z's are on, because you always accept when Z is off, and that screws up your tuning the activity centers well. You want them tuned well for when Z is on. So basically, if you only you only consider the acceptances when the Z is on, then you're going to tune them well for when Z is on, and that you end up tuning them better than if you're just averaging over when Z is on and off. But even there, you can. I guess it becomes more and more challenging the more uncertainty there is in ID, the more variation in how many samples are associated with each activity center across iterations.
Yeah, I'll have to chew on that for a little bit before I fully understand. But
unmarked SCR, it's harder to tune the activity centers because there's just a lot more variation. You would be more likely to be in the case where you might have between like 1 and like 12 samples or something like that assigned to one individual. Whereas if you have, say, all this genotype information, there's very little difference in how many samples are allocated to each individual on each iteration.
Right.
And so like the tuning, the best tuning parameters are, I don't know, it's easier to let Nibble auto tune, and you end up with good acceptance rates.
Cool. Yeah, I'll get on your GitHub and poke around, and I'm interested to see how the guts work. I'll do one last call for questions from folks from online. Just 1 second. Okay, not seeing any trickle in, but so with that, thank you so much for coming, Ben. That was a really nice talk, and yeah, especially the slide at the end with the different flavors of SCR really helps me kind of situate everything.
Yeah, I think my taxonomy there is still kind of fluid. Trying to figure out how, because I definitely, like, if you asked me 1 year ago, 2 years ago, that would have been a lot fuzzier. The relationship of all these different things.
Yeah, it's been fun I follow you on Twitter, so it's been fun to see the evolution. Well, cool. So for folks, give Ben another virtual round of applause, and join us next week for I can't remember off the top of my head who's speaking, but it's sure to be a good one, and we'll send out a reminder email next week.
Oh wait, I got one last. If Sarah Converse is here, or you want to, if she's not, someone who can tell her the reversible jump idea gives. I've been tweeting about this, but it's an alternative way to do multi-session where you can retain the Poisson assumption without the trick that her and Andy used in that 2014 paper. It seems like a more natural way to do multi-session SCR with reversible jump, so you can keep that in Poisson assumption across the sessions. Anyway, that's one last thing I wanted to say.
Awesome. Yeah, well, I'll share that with her. Thanks so much, Ben. And yeah, again,